Everybody, my name is Murad. My mentor is Dr. Isquith, and today I will be presenting my research project, which was focused on studying the effect of high-intensity magnetic field on the stem cells in planetary regeneration. So before I start the presentation, I would like to go over the outline of the presentation to give you a quick overview of the things that will be covered in the presentation. First of all, I want to start the introduction to give you some general information about planaria and why they are used as a model organism in regeneration studies. Then I will go over the background and literature of the presentation, which will discuss some of the most recent findings in the field of stem cell mediated regeneration. After that, I want to talk about the purpose of the study and why we decided to conduct this study specifically. Then I will explain the procedure we followed to achieve the goal of the experiment. After that, I will go over the results of the experiment. And then finally, I will conclude with a discussion of the findings of the experiment and then the research conclusion. So moving on to the next slide. The next slide is the introduction. So I will now explain some general information about planaria and why they are used as a model organism in stem cell mediated regeneration studies. First of all, planaria are free living flatworms. They are members of the phylum Platyhelminth and class Turbularia. Second of all, planaria live in fresh water under rocks. They like to stay in dark environments because they are light sensitive. Third of all, planaria are widely known for having a fascinating ability of regenerating lost parts and organs. This fascinating ability of regenerating lost organs and tissues allow them to be a model organism that has been utilized in numerous studies focused on regeneration, such as stem cell therapy and regenerative medicine. Planaria regeneration depends on the activity of a specific type of stem cells that are distributed throughout the flatworm's body. These stem cells have a special name, which is nooplasts. Nooplasts compose 25% of the stem cells found in planaria, which explains why they're good at regeneration. So moving on to the next slide, which is the background of literature. So we noticed that most published research studies are focused on the effects of extremely low-intensity electromagnetic fields on planetary regeneration. These low-intensity electromagnetic fields are usually generated by a Helmholtz coil. And I have attached a picture of a Helmholtz coil. As you can see in the picture, there are two coils which are attached to each other by a plastic rod. And then these two coils are connected to a power source. And then there's a stage or holder between these two coils, which is used to support the sample, which is planaria in this case. So the findings of these research studies show that low-intensity electromagnetic fields stimulate regeneration. Moving on to the next slide, which is the purpose of the study. So we were interested in studying the high-intensity magnetic field because we could not find any published research studies that were focused on studying the effect of a strong magnetic field on planetary regeneration. What I mean by a strong field is any field that is above 5,000 Gauss. Therefore, since most published research studies were focused on the effects of low-intensity electromagnetic fields on planetary regeneration, we decided to focus our study on high-intensity magnetic fields. The main focus of our study is to determine the rate of planetary regeneration when they are placed in a constant high-intensity magnetic fields. Moving on to the next slide, which is the procedure. So planaria were cut in half, as you can see in the picture here. This resulted in a head and a tail for each planaria. Then baseline measurements were taken by measuring the length of the tail and then the length of the head of each planaria. After that, we placed these heads and tails in small petri dishes. These small petri dishes were then placed in a constant high magnetic field. And I've attached a picture of the magnet that's used to generate the high intensity magnetic fields. So we placed the petri dish between these two poles right here. Now I want to talk about the trials we conducted. So we obtained about 60 planaria and we did 4 trials. Each trial consisted of 10 planaria. 5 of these planaria were placed in the control group, while the other 5 were placed in the experimental group. Okay, moving on to the next slide, which is the results. So the result shows that the average growth of the heads in the magnetic field is 43.2% less than the control group. Similarly, the average growth of tails in the magnetic field is 42.6% less than the control group average. And these results are summarized in this graph. So the average growth of heads in the control group is 0.76, and the average growth of heads in the experimental group is 0.49. So we can see that there is a clear decrease between these two groups. Similarly, the average growth of tails in the control group is 0.74, and the average growth of tails in the experimental group is 0.48. So there's also a clear decrease between these two groups also. So moving on to the next slide, which is the discussion and conclusion. So the results for studies show that high intensity magnetic field decreased growth. In contrast, it was reported that low intensity magnetic fields increase growth. So there's a clear difference between these two types of magnetic fields. One of them increased the growth rate and the other decreased the growth rate. So we know that the accumulation of free radicals is important to initiate regeneration. Since low intensity magnetic fields promote the formation of free radicals, regeneration is accelerated and increased when planaria are placed in a low intensity magnetic fields. In contrast, since high intensity magnetic fields promote the recombination of free electrons, regeneration is slowed or decreased when planaria are placed in a high intensity magnetic fields. So these are some of the findings of our study, and these findings are summarized in the next slide. So this is a graph that shows some of the findings. So we, as you guys can see from the left side, this is the parent molecule, and it has two electrons. When these electrons are excited, they dissociate. If they are placed in a high intensity magnetic fields, they form a singlet state. And these electrons recombine, which leads or prevents the formation of free radicals. On the other hand, if these electrons are placed in a low intensity magnetic fields, they form a triplet state, meaning that they separate, leading to the formation of free radicals. And since we know that free radicals are required for the initiation of planaria regeneration, we can conclude that the high intensity magnetic fields lead to a decreased growth rate. So yeah, that's my research project. Thank you for listening.